Hey guys, welcome to Biomics. In this today's video, we are going to discuss about the 5' capping of mRNA. So what happens in case of a prokaryote? The process of DNA replication, transcription and translation takes place in the cytoplasm. But in case of eukaryotes, the process of DNA replication and transcription, that means formation of mRNA, takes place in the nucleus. So for the formation of proteins, it is being transferred into the cytoplasm. So for this, it requires some modification of this mRNA like 5' capping and 3' polyhydrogenation, due to which it can be transferred into the cytoplasm. It will have a signal to transfer into the cytoplasm, as well as it will protect the mRNA from degradation from exonucleases which are present in the cytoplasm. Okay, then it will help in the formation of protein synthesis, that means translation. So let us see in this video how the 5' capping takes place in the mRNA. So there are three phosphates, that means alpha, beta and gamma phosphate in the nucleotide. It is in the form of triphosphate. So there is a enzyme phosphatase which works here, that is known as RNA triphosphatase. In this triphosphate, what happens in case of a mRNA? The formation of mRNA takes place from the 5' end to 3' end. So the starting is the 5' end. Okay, here there is three phosphates are present, and this adenine, it is having three phosphates like alpha, beta and gamma. By the help of RNA triphosphatase, the alpha phosphate of this adenine is being removed. So here remains only the beta and gamma phosphate. Beta and gamma phosphate. This beta phosphate, what it does, it attacks with the alpha phosphate of guanine. Okay, so here the beta and gamma phosphate are being removed, and it forms a bond between 5' of alpha, 5' of beta, 5' beta and gamma of alpha of adenine, and guanine, it is having only the alpha phosphate. So the bond is formed between 5' alpha of guanine, 5' beta and gamma of adenine. Okay, so there is a bond between 5' to 5', but in general there is a bond between 5' to 3'. In the 5' capping, there is a bond between 5' and 5' over here. Okay, what happens next? The methyl, there is another one enzyme which is known as methyl transferase. What it does, it will transfer the methyl group. Where it will transfer the methyl group? In the guanine. In the guanine, in the 7th position of guanine, it will transfer a methyl group. So this is known as 0' capping. This is known as 0' capping. If the methyl group is added in this adenine, in this adenine also methyl group is being added, so it is known as primary capping. Okay, if adenine is not present in the N group and in the N number, if the adenine is not present, it will be again methylated in the second nucleotide. If here the second nucleotide is being methylated, it is known as secondary capping. This is how the methylation or the 5' capping of the mRNA takes place to protect the mRNA from degradation from certain exonucleases. As well, it is responsible for the transfer of the mRNA from the nucleus to cytoplasm, due to